Thanks everyone for listening. My name is Abhi. I'm a machine learning research scientist at Arc Institute, where I work on our virtual cell models and perturbation response prediction models. Today I'm joined with two esteemed professors, Dr. Hani Goodarzi and Dr. Wang. Hani is a professor at UCSF and a core investigator at Arc Institute. His work spans across the stack of biology and machine learning. So his group is involved in the EVO models here, the state models here, which focus on transcriptomic data, codon FM, so he does a lot of work in RNA biology, and then also stuff as pertaining to the wet lab. He also is the recipient of the Vilcek Prize and the AACR MPM Transformative Cancer Research Award, as well as the NIH Director's Transformative Research Award. So Hani, thank you for joining us today.
Thanks for having me.
And then Dr. Wang is the Senior Vice President and Head of Biomedical AI at Xaira Therapeutics. Somewhat new company, not super new. He's also a professor at University of Toronto, the Chief AI Scientist at UHN, or the University Health Network. And he also holds a position as the CFAR AI Chair at the Vector Institute, which is an esteemed AI institute. Many listeners will also know his work and his group's work on SCGBT, on spatial transcriptomics, and really all things virtual cell related. So Dr. Wang, thank you so much for joining us today.
Thank you for having me here.
Awesome. So today we're going to be talking a little bit about the BioReason series of models. There's many models, I guess, reasoning models that build on top of language models that have kind of been hyped up. I guess the trajectory of these really is from 2023, when people were just telling models to think step by step and, chain of thoughts. And then 2024 comes around and we have O1 preview from OpenAI. And these essentially use reinforcement learning to get the models to do reasoning before they predict the next token. And so BioReason is a series of models that Bo and Hani co-supervised that apply these reasoning models in different domains in biology. So maybe could you guys talk a little bit about what is BioReason 1 and what kind of architectural improvements does it use, and what is really the domain application of this model?
Yeah, sure. I can start. So BioReason 1 is really the first attempt for us to apply the reasoning language models to the domain of biology. What we specifically did for BioReason 1 is we connected a DNA foundation model, which is specifically EVO2, developed by this very Arc Institute, together with another reasoning language model, which is Qwen3, which is a very popular open source reasoning language models. And we really use these reasoning traits and teach language models that are trained on lots of free text to understand biological modalities such as DNA sequences, which were never trained during the pre-training of these language models. And we also use similar techniques such as reinforced learning to really teach the language model to think a bit, to understand all the DNA sequences, so that these language models can interpret tokens from DNA sequences and start to answer questions about whether there's a mutation or what's other biological downstream effects from these DNA sequences.
And so I guess the analogy is like a vision language model, for example, can answer questions or reason with images and text, and then BioReason is able to look, quote unquote, look at DNA sequences and answer questions about them.
That is correct.
Yeah, I mean, actually, practically speaking, that's exactly what we use. So Qwen3 has a vision module, and we piggybacked on that for inserting EVO embeddings into Qwen3 for training. One thing that I would add to what Bo mentioned, I think it's fundamentally important to think about like the strengths that each of these modalities bring, right? So we and others have put a lot of effort into training biological foundation models. And these models are, again, biological. So they have kind of trained on biological data and can think through abstractions of biological data structures. And then we have language models and reasoning models that have a part of world knowledge and also abilities to kind of do reasoning traces, right? Think through step-by-step a process, which is very human-like, and that's what we appreciate about them. So fundamentally, I think putting the two of them together makes a lot of intuitive sense, because that's actually how us as humans interact with biology and biological models. Kind of to give a broad example, let's say, I say there is a variant, a human variant that, EVO2 or some other Genome Foundation model gives us this variant as so-so score. It's kind of on the line, a threshold of what is significant and what is not. And then I just also turn back and tell you, oh, by the way, this is also in P53, right? So just the mere knowledge of the fact that this is in P53 tells us something. We already know a lot about the context of this protein. We know how important it is in cancer. We know it's, mutations in this, like null mutations in this protein is like a very important tumor suppressor and all that. And just knowing that context, us as humans, basically allows us to elevate that variant from that mathematical threshold that EVO2 assigns it to something much higher. Now, whether that is warranted or not, that's, I think, a different conversation. But that is basically the way to really the framework to think about it, that there's fundamentally this broader knowledge and a different approach to thinking about biology that I think biological foundation models don't necessarily access. And kind of coupling them with reasoning models actually allows us to kind of copy or emulate how humans actually talk and take advantage of these models.
And I guess, for example, EVO, taking the example of EVO, is trained using a self-supervised objective. So just predicting the next nucleotide. It's not really trained for these like question answering purposes and understanding variant effects. And so training this reasoning model on top of those embeddings, in the case of BioReason 1, I guess helps the model tweeze out that and is more, I guess, applicable for researchers.
That is correct. Just really want to echo on Hani's point that combining language model, particularly reasoning language model, on top of the biological foundation model is really a key innovation of this whole BioReason series, in the sense that most of the language model already trained with lots of reasoning data. And we always know that biology foundation models are very bottlenecked by data scarcity, which is true. And really kind of trying to utilizing the strong reasoning capacity of language model. And what we really need to do is to align the tokens from different modalities. Teach the language model what does the DNA sequence token looks like, so that the language start to recognize these very strange or bizarre tokens we've never seen before during pre-training, but start to understand very different biological sequence modalities, so that it can start to marry the reasoning capacity with the DNA sequence information to interpret lots of biological questions.
And I would also want to really emphasize that deep fusion, I think it's really important. I think agents have been quite popular these days, and they have been also popular in biology as well. But I think in the end of the day, agents are as good as the tools they have access to. And going back to Bo's point about kind of data scarcity in biology, I think a lot of our biological models, just by design, are fairly handicapped because they don't have access to enough data, pre-training data. So this is not just simply as deploying them as tools. Right? It's really giving the model access to the abstractions of the biological foundation model, so it can operate at that level, as opposed to simply kind of the output of the model that you would normally interact with as if you were approaching it as an agent.
Can you maybe elaborate a little bit on how do we generate this training data for BioReason 1 before we get into BioReason 2?
I'll let Bo go first.
Yeah. So curating the reasoning data sets is actually one of the most challenging tasks for BioReason projects. We spend probably more than 80% of the time, efforts, resources on just data curation. What we did for different BioReason series is that from certain biological input, let's say DNA sequences or protein sequences, we ask a lot frontier reasoning models such as GPT-5, so that for these language models to interpret these DNA sequences, we also have human experts to verify and improve the answers by these frontier language models, so that we kind of have a relatively high confidence about the quality of the response because, every frontier language model hallucinate once in a while. So with this human in the loop pipeline, we quickly curated lots of reasoning trace for training BioReason series. Certainly, it's not very automatic right now. It still requires lots of manual checking, manual verification. But I do think what the whole purpose of BioReason is not to really replace human biologists. It's really to have a collaborative loop so that the AI can generate the first round of answers, and then human experts trying to verify and improve, and further to guide. We can write better prompts to these frontier language models to generate better reasoning trace. By this iterative process, we start to curate lots of reasoning data for BioReason. I think the good thing about reasoning language model is that we don't need millions and millions of training data to apply reinforced learning. In fact, actually, lots of research says, you actually only need 50 or more, a little bit more reasoning data to use reinforced learning. I think this first proposed by DeepSeek team. And we kind of really take advantage of that. We start to really curate a very high quality reasoning trace, stepwise thinking, and then let the language model, especially Qwen3, to understand how human biologists reason by looking at different biological modalities. And with just relatively small batch of high quality reasoning trace data sets, we can train the model relatively well. Certainly there's lots of nuance about how human kind of cure the small, reasoning data at different steps. But overall, we kind of having a collaborative loop that are combining frontier language model with human experts to generate high quality data sets so that we can apply reinforced learning such as GRPO to train these language models.
Yeah, ultimately, most of it is founded on kind of really well curated knowledge databases, but turning those databases into reasoning traces is something that frontier LLM models are increasingly good at. And again, with kind of human in the loop approach, like let's say, for example, like how do I as a human kind of interact with a protein sequence that I've never seen before, right? So I will like take it, I will look at, like put it into kind of tools that will tell me what domains it has, look at what it potentially interacts with in the cell, and that kind of these kinds of knowledge domains will tell me like what is it that this protein likely do in the cell, right? So kind of emulating this process is exactly what we mean when we say we're creating reasoning traces, right? This is a protein. Again, the protein is represented in this case as ESN embeddings, right? So it's a protein embedding really, and that is associated with these are the domains that this protein has, right? Which are basically annotated, but and then from these domains, for example, if there's a DNA binding domain, it's probably could be a chromatin modifier, could be a transcription factor. So all of these kind of components are possibilities, and the model looks at the context, looks at what other domains are there, predicts what are the interacting partners, and based on all of these can reason around, well, is it a nuclear protein? Does it bind DNA? Is it a transcription factor? And kind of this process is exactly what a human would go through to come up with an answer to the question of what does this protein do, and we have basically tried to simulate that using other language models, and then that's kind of
it's like a bootstrapping.
Exactly, and that's what we use with reinforcement learning and GRPO to train the model further on this kind of reasoning traces.
So actually, that's a good segue in that. So for the listeners, BioReason 1 was a model trained on reasoning over DNA sequences with the EVO model. And BioReason 2 or BioReason-Pro is the latest and greatest BioReason model. It's trained for all sorts of question answering about proteins, right? So could we discuss, maybe I would love to get your thoughts a little bit on what are the problems that BioReason-Pro solves? And we talked a little bit about how does BioReason differ from these clinical models and what applications are there. I think a lot of listeners would be familiar, for example, with AlphaFold as another way to probe sort of what is protein structure, function, So what does BioReason-Pro excel at? What, through the development of it, what are you guys excited about?
Yeah. So the problem BioReason 2 or BioReason Pro trying to solve is really a huge gap of annotation problems. That is, we have lots of protein sequences, and we don't know the functions of these proteins. And as you mentioned, AlphaFold is really award-winning solutions that takes the sequences we're trying to predict the folding structures. And this is largely a solved problem. However, there's a next step, which is, can we know the functions of these proteins from the sequence alone? And there's a huge gap to really take the sequences to understand the functions. And having any models, AI models that can take the sequence and predict the function is of huge values to drug discovery process. And what BioReason 2 is designed is to really as an early attempt to fill the gaps of function annotations.
Actually, one thing that was very interesting that came out of this effort was kind of the realization that actually to what extent our knowledge is finite, right? Even when you say, we have a curated database like Go terms, for example, or you put a protein and you get all the Go terms. But somewhere along the line, we realized that the model goes much further than where the Go terms are. So you have this hierarchy, right? So it goes deeper into the tree in a very reasonable way. So it seems like the biology hasn't really caught up yet. So you will penalize the model for doing this, but in reality, the model is actually going further than the human curation has gone, which actually could possibly be the correct answer, actually.
I guess we don't know if it's hallucinated or
exactly.
novel
So
biology.
yeah, so there are a couple of things. One is there's temporal separation, so you can use some of the data and see things that came later, how did we do there? But also actually human evals, right? So actually asking humans to go through it and see whether that makes sense. Is it truly hallucination or actually could be the right answer? The bottom line is, again, as Bo mentioned, the goal of these models is to be hypothesis generators, right? And the place that I think this can have kind of huge impacts is, as we go deeper and deeper into the sequence space, be it, in genomics, for example, where we're sequencing a ton of genomes and a lot of coding sequences, especially in species that sometimes don't really have a lot of homology-based kind of annotation calls. I think have a tool like this that not only can predict what the potential function is, but also kind of walk you through its reasoning. I think it's incredibly useful as a tool, or in kind of synthetic biology applications that you want to kind of keep an eye on. This is the function that I want to for my protein to have, and now as I'm optimizing other factors, I want to keep that in mind. Again, having these kinds of models are incredibly useful. And finally, I think that the reasoning itself is sometimes quite helpful, and it's just how the model is actually thinking through the process. I think quite illustrative as well.
I really agree what Hani says, that having a reasoning model is so much better than just have a prediction machines. Most of the time, people can train a classifier that says what's functions. We formulate as either a binary problem or multi-class problems. But what eventually a biologist gets is just a number. I think what BioReason-Pro offers is really additional reasoning traits so that biologists can look at it using free text in a way that human biologists can understand. They can either verify or they can generate new hypotheses from reading the outputs of these reasoning models. And they can also, if they disagree or they think it's likely wrong or it's a hallucination, they can actually do similar wet lab experiments to verify it so that they can provide feedback loops to these AI models. It's just so much more rich information than just prediction models.
People talk about interpretability in some of these models. That is
correct. We can actually have a more mechanic interpretations of what these AI models are look at, so that we can understand, just a lot more information beyond just a number or likelihood vectors.
You have these huge repositories of collected protein sequences that we're sitting on. What is maybe a use case that you guys see for the BioReason-Pro model? For example, AlphaFold DB pre-calculates all of these protein structures for these sequences. So do you see this as a tool that maybe can be run over all of these sequences? And then I guess, is that of usefulness to biologists?
Yeah, we actually was discussing, and probably will do it, is that we can generate a similar database such as AlphaFold DB to basically list all the functions for all the sequences that is existing. And I think having those functions available to biologists or to drug discovery scientists will be extremely valuable for them to generate new hypotheses. And that's also exactly our plan, that we will open source all the models, all the training data, training codes, so that everyone can verify, everyone can replicate, so that we can have the biggest impact to the whole scientific community.
Yeah, I think kind of filling the gaps for a lot of curated databases will also be a good application of this. And that's something that we have realized, that, as I said, you look at Go terms, and clearly literature is further along than the Go terms are. So at some point, someone has to update it. So as our knowledge base grows, I mean, that human curation becomes a bottleneck. And actually, we would need models like this to at least orient the human curators that, this is what I think should go here, and you can check that, as opposed to just like waiting for that to happen organically. And we have seen this with Go terms, but that's also it's true for DNA variants and kind of other applications of this type of reasoning models as well.
Have you seen that the model is actually receptive to like single amino acid changes, and can it detect that one protein may be functional, but a very similar looking protein may not be functional?
I think that we actually look at there are lots of poking around of the models. We find sometimes the model hallucinates. Sometimes the model fails to capture very subtle or single nuclear mutations to the final outputs. This is understandable in the sense that we don't have too many of the reasoning traits yet, and the accuracy of sequence to functions, although it's state-of-the-art, but still kind of stuck at 70% level of accuracy. So there's still a gap for us to fully understand what's the connection between sequence and functions. And therefore, sometimes there's edge cases where you mutated the sequence, the model simply just didn't find any difference of the input, and therefore they have very similar outputs. So I think that's definitely a point of improvement for future development. But we really want to put out the model to broader audience community so that they can help curate more training data to further improve the model.
And I guess as even the embeddings of the protein and amino acids themselves improve and are more receptive to these kinds of things, the model benefits for free.
Yes, if we have a better sequence embedding models, certainly our BioReason-Pro will benefit from that too.
Yeah, we know that we can do supervised fine-tuning on, say, like deep mutational scanning data, right, of that sorts, and with biological foundation models. So the information is definitely there, and the goal would be to connect it to kind of a functional understanding. And I think, again, given enough reasoning traces of that type and enough training data of that type, I think these models can actually get there. It's just that now it's a much more coarse-grained kind of domain level understanding of like protein and sequences and functions. Like we have really brought this again domain centric view to protein function. Like these are the domains the protein has, and like all of that is learned from the embedding, and then the function is reasoned through that. So unless the change is so obviously breaking that domain that the model just decides this doesn't exist anymore, it would be actually, in the current resolution, it would be actually a hard problem to solve. But I think if we show many examples of kind of deep mutational scan type or variant effect type data sets to the model where we actually show small changes in the embedding space, how it can impact function, and along with the reasoning trace that goes through it. And for example, oh, this variant is right in the middle of the like an important part of the DNA binding domain, right? So this should abrogate its function as a transcription factor. I think this kinds of, again, going back to what I said earlier, that I think it really all comes down to how well you can put together these reasoning traces. And I think that would be kind of the biggest limitation in the end.
Great. So, I mean, I'm curious about the future of BioReason models. So BioReason 1 was DNA, BioReason 2 was protein. Are we going to see models reasoning across these scales? Are we going to see clinical models? What's next?
That's a great question. So we're already starting project of BioReason 3, which is trying to use reasoning language models to interpret cellular foundation model, or in other term, virtual cell models. I think that's very exciting for myself, since we developed one of the early foundation model in single cells. So we want to see how reasoning language model can help enhance or interpret these single cell foundation models. And connecting cellular biology to clinical science is certainly very, very interesting to me as well. But there's a huge gap from cellular models to human models. And having models such as BioReason, that's actually will open lots of opportunities to connecting these cellular models to clinical models. So definitely very interesting in that direction. The bottleneck will be enough paired high quality data from patients to cellular models. So, but using really these powerful frontier reasoning language models, we are quickly kind of providing lots of new capacities for us to model a patient response to certain interventions.
Mm-hmm.
Yeah, I would say along those lines, I think kind of thinking through how we can build multi-scale models as well, right? So you have a cell, the cell is made up of DNA and protein and all that. So how can you kind of link these modalities? For example, if I say I'm going to mutate this amino acid in this protein, what is the impact of it on a cell, right? Reasoning through all of that, starting from a protein embedding and going to cell embedding. I think this is kind of the future of thinking about BioReason models. But again, where we started, right, 80% of this effort is building the training data.
Right.
I think that reinforcement learning is quite capable. And as we are doing this, I think people will come out with other ways of doing this, with even fewer examples and or more efficient paths. But again, we have to really focus on kind of where the quality of data exists, where we can actually get our hands on enough reasoning traces to be able to train well.
And I think there's a point to be made too about like the verifiability of the scale that you're at, right? Like at the molecular level, we can now generate DNA sequences, and I mean we can generate amino acid sequences. We can do some of these deep mutational scans that you were discussing. But already once you get to the cellular level, like how do you design a cell with like this specific gene expression value for one gene and this specific, like it becomes very difficult to actually engineer the starting state for some of these. And then, the level above cell gets even more difficult. So I think that's also an interesting question, it seems like.
Yeah, I mean, both the data, but again, like at some point we even run out of, because like we simulate these reasoning traces around like how us as humans think about them, like think through them. But again, our capabilities are limited. Right? So at some point we even like run out of ways of thinking about it. Like how would you even do this, like as a human? Right? So if once you hit that wall, I think we have to really rethink what it means to have reasoning models and what it means to have a reasoning trace. I see a future that hopefully the model can also figure that out on its own. But of course, at the cost of maybe some of the interpretability and us being able to read through the reasoning traces in a kind of comprehensible manner. But to me, that aspect of it is extremely important. Right? So it's not, again, the goal of this for me is is the language component of it as much as the predictive capabilities. I think being able to kind of get a sense of how we got to this answer, even if at the same, or even like lower performance, I think it's extremely valuable.
Mm-hmm. Have you guys seen any like adoption of the first BioReason, or are you guys using these models at all in your own groups?
Yeah, we use BioReason Wang, which start to augment lots of DNA language models, and now we have BioReason Pro that really add lots of new tools to take the protein sequences to understand more rich context information from the function space. And now we're also interested in extend this BioReason to virtual cell space, where you can start to using languages to interpret other cellular, for example, single-cell RNA-seq data sets. And eventually, I think these reasoning language model will really revolutionize the whole space of biomedical research, where not only we can use this language model to do literature survey, but also we can start to develop agentic frameworks or more reasoning, multi-agent system trying to mine these data sets automatically and generate hypothesis, summarize what we can find, summarize what's the future state looks like. We'll provide lots of really valuable insights for many biological discoveries.
And also giving a quick shout out to the students who have actually done all the work. They're first to tell us how many people are, following the GitHub repos and
That's awesome. Yeah.
downloads and all that. So, ultimately it's been quite impressive to see kind of how large some of these teams need to become to hit a critical mass of, people working together. A problem of this type that has so many kind of angles to look at it. At some point, we even had like competitions between architectures, and this architecture would be better or that architecture.
You made the best one right now.
Yeah, exactly. So I think ultimately, again, it's more about just kind of us learning from the models as much as the models learning from us.
And yeah, like on the point of agents, like once you train this model that can do, in the case of BioReason-Pro, sequence to function, suddenly you have like a very powerful tool in the toolbox of these larger models, being developed by frontier labs. Really, the whole process is kind of like self-accelerating, it seems like.
That is true. That is true. So the only BioReason-Pro can serve as one of the tools for agents to call, but because we have reasoning capacities, we can actually evolve different agents from these reasoning language models. So I think it's actually very exciting research direction to train different agent systems that it can evolve from these language models, so that it can trying to critique, trying to verify from each other, and have a collective intelligence from these reasoning language models to better the prediction for different biological tasks.
I think going back to Bo's point, I think that's kind of where the power of these models are, and hopefully we can turn them into agents in a not too distant future as well.
Yeah.
Awesome. Well, folks can find BioReason 1 and BioReason Pro on GitHub, fully open source data as well. So thanks again so much, Dr. Hani Goodarzi, Dr. Bo Wang, for your time today.
Thanks, Abhi, for taking the time.
Yeah, thanks for the listeners.